Afternoon, everyone. Thanks for joining us for another installment of Intellectual Journeys. It's a great pleasure today to welcome Professor Caleb, better known as Tuck to all of us, Finch, who is a university professor and the ARCO William F. Kieschnick Chair in the Neurobiology of Aging, and Professor of Gerontology, Biological Sciences, Anthropology, and Psychology. Those titles, I think, give you an idea of the amazing spread of his interests and activities. He got a BS in 1961 at Yale University, a PhD at Rockefeller University in 1969. He came to USC in 1972, became a full professor in 1978. His research, at least in part, let's say, is centered on the biological mechanisms of aging. But that general description, I think, encompasses a whole lot of topics that I think he will give us a more detailed look at in a minute. He's the author of 6 books and the editor or co-editor of many more. 600 articles and abstracts, and I think still counting. He also is an expert on Alzheimer's disease, and he founded the Alzheimer's Research Center in 1983. His recent research has also concerned the connection between urban air pollution and Alzheimer's disease. So Tuck, thank you very much. We're looking forward to hearing from you.
Okay. Well, I first started to think about aging, close to 60 years ago, in thinking about what I wanted to do for my PhD project at Rockefeller. And basically, there are 3 core questions. I look at aging processes the way developmental biologists do, that there are changes in gene expression that are cell specific, and that perspective had not been brought into the field when I began to think about it. And I look at gene expression and aging from a cellular level to population demography levels, and I'm interested in how aging differs by environment, human aging, and how does human aging differ from other species? And all of this is seen in a context of molecular biology, biochemistry, and that has from which I've gotten very specific experimental approaches. So I came to USC in 1971. First meeting was with the vice president provost at that time, Zorab Kaprelian, who to whom I was recommended by Ed Tatum, who is a mentor of mine at Rockefeller, and Jim Biren, who was recruited to USC in 1968. And Zorab was extraordinarily important because he was essentially building USC to a new level, and it's his remarkable leadership in the School of Engineering that brought us into the top 5, where we've been ever since the mid-70s. And he recruited specific people across sciences and humanities and arts. And Jim Biren was one of his recruitments. Jim was a psychologist, one of the pioneers in gerontology. And he said, if you ever have any problems with Jim, give me a call. Well, Jim was extremely supportive in the beginning, but then as I began to be interested in Alzheimer's disease, he was unhappy about that because he said, you're converting aging to disease. And I'm interested in real aging, not disease. And I said, that is not how the biology of aging works. The longer you live, the higher risk of mortality because of accumulated lesions, cancer, heart disease being among them. And as I will tell, he couldn't say no when I brought in a large amount of money with the Alzheimer's Center. But these were, it was an unusual recruitment to start with the second most powerful person in the university, who was, we had a dinner and a breakfast, and there were a couple of other conversations in my visits. But he convinced me that USC was a place that was going to be on the map, and it was really exciting to be part of that early discussion. And a couple of other people that here's Jim Biren and Margie Gatz, who I will say more about. And I can't possibly go through the wonderful number of people that I've trained and who've trained me reciprocally, but we've had a heck of a good time within the lab and with our collaborators, and I'm really happy. But let me mention a few people that are particularly important over the years. Ed Schneider, who I recruited as a dean, he was the deputy head of the NIA, and he brought an immense energy and insight in recruiting biological faculty and demographic faculty. And Mike Waterman, of course, who's been a great figure at USC and who gave one of the first emeriti talks. Myron Goodman, who I helped recruit in 1973, and they have been extremely important intellectual colleagues. And Henry Antosovitz, who was in the math department, wanted to be part of the biological discussions. And then three colleagues in particular, Rich Bergman, who is here on this Zoom, has been a shoulder-to-shoulder colleague and for 35, maybe 40 years, and just a superb scientist. Eileen Crimmins, with who I continue to work, and a deceased colleague who was extremely important, Richard Thompson, who really built neuroscience at USC. Along the way, I've written some books, and I write them not for any other purpose than I want to understand the topic that I'm writing. And each one of these has been a change in my vision of what I thought I was studying and why. And the three themes that I posed at the beginning were essentially right now what I think I've thought about. But as we all know, memory at each point is reconstructed, and one cannot ever be absolutely sure that that was the reason you were thinking about something at an earlier point. So in the beginning, first decade, I was recruited to set my lab up in 1972. I was the only neuroscientist at USC for 4 years. And it was pretty lonely because people didn't understand why I was working on the brain, because it was too complicated. You can't do molecular biology on the brain. And what is wrong, of course, you can. And I wanted to do both work on mice as well as on humans. And so I got the idea of with an assistant professor at the Karolinska, Bengt Windblad, we put together the first post-mortem brain bank of normal aging. And that took about 4 years. And from that, we were able to see some things that nobody really anticipated, that normal brain aging begins in middle age in humans and mice with losses of synapses, but without any pathology. And here is on the top row, the dopamine receptor D2 subtype showing a decline from 3 to 8 to 20 to 28 months, which is across the mouse lifespan, and parallel data in humans 20 to 80 plus. And we had a really hard time getting this published because for 3 years, serious journals said, but these must have pathology. And finally, I brought in enough evidence from psychology that there was exactly parallel change in cognitive processing and in the speed of cognitive processing. Jack McArdle, who was at USC in the 90s and 2000s, who's now died, did a superb work to document this, as did Jim Biren, but it's very clear that the brain functions differently at 40 than it did at 20. You can't remember as long a string of numbers, and your ability to handle multiple cognitive processes at the same time declines. And so all of this is totally separate cell biology from dementias, which happen increasingly after the age of 60. So this was really a major effort on the first 10 years that then put me in a position to begin to think about the Alzheimer's programs. And the new director of the NIA, Robert Butler, in 1983, was talking around the country about a program of Alzheimer's centers, and UCLA originally said, well, they were going to do it because they had a much bigger neuroscience program. USC didn't have any, really. It was me and another person or two, Les Weiner and Joe Vandermeulen, who had some work. And so UCLA was going to be the leadership in the proposal for USC and UC Irvine, which had a very strong neuroscience program. So they started work on this in November of 83, and the grant was submitted, getting ready to be submitted. In 6 weeks before the grant was due, they called me up, they called Carl Kottman up down at Irvine saying, geez, sorry, there isn't really enough money to do anything with 2 other universities. We're going in by ourselves. And I went ballistic and put a really good team together from Caltech, from City of Hope, and from UC Irvine with Carl. And essentially, we came in 4th in the country, a couple of 100th points below Hopkins and Harvard. UCSD was the strongest, and Mount Sinai in New York, and UCLA came in 26th. So that was an immense change in my approach to things, because it was now gave a platform for developing a multidisciplinary approach to Alzheimer's disease as well as to neuroscience. And the university trustees, and I forget who was president, I think it may have been Jim Zumberg, said, well, we really got to get some funds going on this. And they recruited Richard Thompson, who I'll show in a moment, I did already, to then build a neuroscience program. And so we now really are on the map. Roughly 100 neuroscience faculty who are funded, and a couple of 100 pre-docs, 4 NIH training grants. And we compete favorably for graduate students with Caltech and Stanford. So I'm really happy. And the person who's running this was Helena Chu. This is shown a few years ago. And here was the original group of us in the LA Times award, picture and an article that there was an Alzheimer's Center at USC now. So that gave a whole new range of resource opportunities. And I had a merit award that supported, for 7 years, supported training of assistant professors and postdocs. And we made a number of findings. I also did a sabbatical at Caltech to learn hands-on molecular biology and brought back some terrific postdocs. So one of the things that was not known at this time was whether you could get messenger RNA out of the postmortem brain. And it turns out, and we showed this, that it's remarkably stable in a mouse brain and in a human brain for up to 48 hours. You can pull out full-length poly-A messenger RNA and make libraries. And that was a seemingly minor publication in 1987 that gave the rationale that you could actually do molecular biology on postmortem brain. So to summarize really 30 years of work in these 4 lines, we and others pulled out of the brain messages that increased with Alzheimer's above aging, and many of them had to do with inflammation and proteins that interacted with the amyloid peptide. And I'm not going to give a long story on that, but there's something like 50 papers that have both mouse in vitro, in vivo, and human comparisons, and that it really shows that inflammation is a fundamental part of brain aging, as well as additional trajectories and pathways of inflammation that are associated with neurodegeneration and Alzheimer's. We also, I'm a physiologist, taught physiology with Rich Bergman and in the Alzheimer's disease. And now it's well understood that we had the first paper on that in 2003 or something like that, no, 98. And it's now much more understood that Alzheimer's has a huge systemic component and is not just restricted to the brain itself. We also studied how some of the proteins that are increased with Alzheimer's disease alter the toxicity of the amyloid peptide, and found a new biophysical organization called oligomeric peptides that are more toxic than the monomer or the neurofibrils. And this led to collaborations with Abbott and the formation of a drug company that's now on the market, Acumen Pharmaceuticals. And after it took 30 years and careers of roughly 100 people to get a phase 1 drug, monoclonal, which is now in phase 1, ACU-193, being tested in a toxicity clinical trial. And a number of times during the development of Acumen, I was primary founder of it, and USC for a while got some income, but then to avoid patent conflicts with other individuals, a story by itself, I'll not tell in any detail. My original patents with USC were revised sufficiently that USC or me will not get any income from the sale of this drug if it goes anywhere. But along the way, I could have had a second laboratory supported by Acumen. We had a $100 million contract with Merck, and I decided I didn't want to run 2 labs. Running one lab was all that I could do on 724, and I kept my focus at USC. I have to say, as the program developed, I had opportunities to go to 3 different drug companies, and Duke, Stanford, UCLA were interested in recruiting me at one time or another. And I've stayed at USC because of the high level of collegiality here and the relatively small number of sense of competing labs. There was none of that at USC. It has, from the beginning, been extremely receptive to anything I wanted to do. There was nothing you couldn't do at USC. There was no precedent for this is only, we only work on certain kinds of things here, which is very much the case in the more established neuroscience programs. Each of them is known for 2 or 3 areas that they're very good at. And you want to sort of be in one of the clubs. Otherwise, it isn't, you don't get the students and you don't get the administrative support. USC has been the other side of that coin. Things that even didn't have a particularly large funding draw were still of interest to the deans and the provosts that I talked with. So it has been a huge pleasure going from a discovery in my lab of protein organization to see a drug finally on the market. So an important part of all of this, in my thinking, it came with a recognition that there are shared inflammatory processes across organ systems in aging and Alzheimer's. And here is something I put together that nobody had seemed to think of, which is comparing the proteins and processes in the plaques on blood vessels, so-called atheromas, with a senile plaque. And to begin with, they have the same monocytic cell, CD68 macrophages, which are foam cells in atheromas and microglia in senile plaques. And there are amyloid proteins of a different sort in atheromas as well as in senile plaques, and inflammatory factors, complement and cytokines. So even though the plaque and the senile plaque the atheroma may look quite different, it turns out that they have a huge shared commonality of inflammatory processes that are ongoing. And within this discussion are whole bunches of papers that take this much further. So in 1997, Floyd Bloom, the editor of Science, asked me to write a review on the genetics of aging. And I said, Floyd, I'm not a geneticist. Why are you asking me? He said, that's why I'm asking you. Find somebody to write it with. So I picked Rudy Tanzi, who's now a major figure in Alzheimer's genetics at Harvard. And what we found was shocking, but extremely strong data, starting with identical twins, that the heritability of lifespan is really very modest, whether it's identical and fraternal compared, or inbred mice, or inbred flies, or inbred worms. It is under 50% and typically 15 to 30%. I mean, just to put a recent piece of data on this, the heritability of identical twins versus MZ versus DZ at age 80 is 12%. So the heritability of lifespan decreases the older you get, implying that there are environmental factors, and I could talk about another dimension. I'm not going to today about developmental variance, but that given the minor effect and penetrance of genetic risk factors for aging, lifestyle and other environmental influences may profoundly modify outcomes of aging. And in the last 10 years, I've helped develop a program. I say helped because nothing I do is by myself. It's with colleagues who put major effort in with me. We have defined the emerging role of air pollution in dementia and aging processes. So the idea of aging being environmental came from an article that I wrote for Science in 1990 and a follow-up a year ago, which looked at the rates of mortality increase with age. And here is the deaths per year in Australia and the United States. And what you see is on a log scale that mortality increases roughly on an exponential way. And in one case, some prisoners of war by the Japanese were observed to have, in later data, essentially the same curve shifted up by nearly tenfold. So this gave me the idea that there was a basal process of aging in many environments that followed a remarkably robust law. And in fact, in biology, as we all know, there are very few laws that seem to apply to multiple species and multiple environments. There's sort of laws of electrophysiology, the Hodgkin-Huxley equations, but there are very few things that seem to be general across human populations. And this is described by the Gompertz mortality law, which is an exponential with a coefficient that I defined in great detail for number of species and populations. And the equation predicts that there will be a maximum human lifespan of about 120 years, given this exponential rate of mortality increase. And of course, that is the observed maximum. There is one person, Jean Calment, who is convincing to have lived more than 120, but since she died in 1997, there has not been a single person since who's lived over 120. So the human aging in multiple populations is described by this. Then I got Eileen Crimmins interested in this. She's a demographer. I showed her face earlier at the School of Gerontology. She's been a wonderful colleague, analytic thinking and insight into populations. And we examined the historical populations in a number of countries for their mortality curves across the lifespan. And of course, here's Sweden in 1751 with very high mortality from infection. And here it is, the birth in 1931. And essentially, the entire mortality curve is dropped down, but the exponential slope has not changed. So there's many, many further examples of this, but the environmental effect is very strong, but it does not seem to have materially influenced the exponential slope beta, which reflects the accumulation of tissue damage, cancer being one of the major contributors, of course, atheromas in the blood vessel being another. But it's been great fun to work this out. So taking this a little further, looking at species with many, many different lifespans, in my first book, 1990, comparing mortality rates in a number of different species, I began to think that there might be something called negligible senescence in which for a given species, there might be extremely low levels of mortality change with age that couldn't actually be measured. And so we have real numbers for that, and I'll show them in a minute. But the idea that there is you can describe the comparative biology of senescence in terms of mortality rate accelerations is something that has been found useful by many of my colleagues. And so last year, or the year before, Steve Ostad, who's a wonderful biologist at University of Alabama now, and I summarized what was known for air-breathing vertebrates for longevity and for the Gompertz constant. And mammals have the broadest range of sorry, this is unstable here, of Gompertz constants that go from less than 0.1, which appears to be the naked mole rat, which a little mouse-sized creature that lives 40 plus years and is a candidate for negligible senescence, to some other mouse-like species that have a 1 year lifespan. But you look across the different aerobic vertebrates, and you can see a similar range of mortality rate constants and lifespans. So essentially, you couldn't, within any of these groups, you couldn't tell the difference at a cellular level. A mouse cell is the same size, a blood cell or lymphocyte is the same size as a human. It obviously, there's another level of genomics that has not uncovered what allows lineages that are anatomically so similar to have this range of lifespans evolving in the course of 100 million years. So that's been really fun to put together. And of course, the question is Alzheimer's disease. And here's primate phylogeny, lifespans of very different length. Here's lifespans. The mouse lemur is the shortest lived, maybe 10 years. And it has Alzheimer's disease-like change, including loss of neurons. The chimpanzees and the gorillas have some changes, but they're definitely not Alzheimer's disease. And then humans, which live another 30 years than the great apes, have Alzheimer's, and as I will show this, in some populations, but not others. So my interest in infection and inflammation led me to join a group of anthropologists who are studying lowland Bolivians who have very high levels of infection that cause 80% of their deaths. And yet the heart attacks and strokes are almost unknown. So I joined this and brought in some additional expertise, as you'll hear in a moment, that really has illuminated a completely new set of perspectives on aging. So here is what their population pyramid or mortality rate by decade is. Very high early mortality, and perhaps if you get to age 25, you have a 35-year more life expectancy, and essentially 10% of the population gets to age 60, and there's a very few who get to be 83%, and there's one or two documented 90-year-olds. So a team of cardiologists that I brought in that I'd been working on with looking at possibility of heart disease in ancient mummies, they were astonished. But the reality is, when we actually got data from the imaging that we arranged in Bolivia, it turned out that this group, the Chimane, have calcium in their coronary arteries, but it's accumulated at a rate 25 years slower than in the United States and in Europe. And not only that, when I got Margie Gatz and Helena Chu interested in this, they did a really deep study on cognitive impairment in the Chimane, and essentially there is no advanced Alzheimer's that has been found in the oldest person there. There's mild cognitive impairment at a very, very low level, among the lowest that's been documented. This is a major paper came out, it's actually this year. So they do have mild cognitive impairment, but their dementia has not in any of the cases shown an advanced stage. And this dementia prevalence is among the lowest worldwide. So that was, this is now further documentation of the extraordinary importance of the environment and the outcomes of aging. So this led to an inquiry. This is all in the literature publications. What is the difference between the pre-industrial environment and lifestyle versus the post? So the Chimane, like most people living rurally 200 years ago, have much higher physical activity. Chimane is 15,000 steps a day. And they have low blood lipids, low blood pressure, low blood sugar, in contrast to the broad prevalence in industrial populations. So all of these are physical activity, nutrition, blood pressure, lipids, and glucose are risk factors for cardiovascular disease and Alzheimer's, which raises the question, are these modern diseases? So I've been thinking about this in connection with the help of a wonderful colleague who is at Cal State Los Angeles, Stan Burstein. We've written some papers together. I wrote some on my own, looking for what the ancient Greeks had to say about cognition and aging and longevity. And essentially, we have a paper that's under review right now that suggests that ancient dementia was absent advanced dementia was absent in ancient Greece, but began to appear in the Romans. And this then led to the question is, did lead poisoning have anything to do with it? So we turned up a paper published this year, a description of how the elite Romans liked to sweeten their wine by putting sugar of lead in it, lead acetate, and calculating units. They had essentially a gram of lead per liter of wine that they drank. So there's a possibility that we suggest that advanced dementia is a modern disease. There was it could be induced, and maybe it was present in the Roman era, but the lifestyle and environment, in fact, by comparison with the Chimane, may be actionable preventives in the future by modifying lifestyle. Now, going ahead quite rapidly, I mentioned earlier that I developed with my colleagues an approach to air pollution, which is extremely well documented as a risk factor in heart disease. And Margie Gatz and colleagues here 10 years ago showed in the LA basin different contours of air particle density in association with cognitive learning impairments, memory impairments, and executive functions in home resident middle-aged and older people in the LA basin. So there was a lot that gave the rationale for J.C. Chen, who's a full professor in the preventive medicine, to develop a program. We have a program project that is worth $15 million total that involves USC and UCSD. And essentially, there's 15 really strong papers in the last 10 years from the epidemiological population that air pollution accelerates cognitive aging and causes loss of white matter and affects the frontal temporal lobes, and essentially living in a high air pollution zone adds 1 to 2 years of brain aging, as well as increasing the risk of Alzheimer's by roughly 10 to 20%. Mm-hmm. So experimentally, I was able to approach this with another USC colleague, Sayudis, who devised techniques for collecting air particles and then re-aerosolizing them for mice under precise time and dose. And what we've done is to show that indeed it increases the amount of amyloid in the brain, and it appears that mice carrying the Alzheimer's risk gene ApoE4 are more responsive than the ApoE3, which may be protective in human populations for exposure to air pollution. Here is a study a couple of years ago from northern Manhattan, and comparing ApoE4 carriers for air pollution and for nitric oxide. There's a faster cognitive decline in the ApoE4 on consecutive annual visits of people who are age 65 and older. So there's a gene-environment interaction, and we've done a great deal of work on this at a molecular level, showing the different transcription factor responses to air pollution components and the genes that they regulate that are antioxidant enzymes concerning glutathione as the major antioxidant physiologically. So what's remarkable and satisfying to me is that many of these same genes are changed in transcription during normal aging. So we have a much larger agenda now of systemic aging, tissue-specific and allele-specific aging for a number of genes that have yet to be discovered that modulate differences of lifespan in populations. So the stunning example that's in front of us comes from Eileen Crimmins again, that lower education individuals, high school or less, have roughly 10 to 15 years shorter lifespan than those of us who finished college. And the reason has to do with, and it's not only the differences in lifespan, but here is from another epidemiological group. This scales with the decile of household income or household wealth. So this leads to a huge set of biodemographic questions. And it turns out that there's, of course, multiple levels of explanation, but I want to focus on the developmental side, drawing back on my origins in developmental biology. And I want to point out something that is now known to those in this field, that children from low SES are born biologically disadvantaged. They have anatomically less developed cerebral cortex and hippocampus at birth. There's 10 or 12 papers on that. Their executive function as children is worse. They have childhood obesity, and they are in households where there are more adult smokers and higher air pollution. So this goes down to the molecular level of DNA methylation. I mean, this is so essentially low-income people are at birth are brain damaged, and that has immense implications. So we've developed an animal model for this by exposing mice to air pollution during gestation, and it has a major impact on the adults. The neural stem cells are reduced by more than 50% in the brain, and there's changes in metabolism. They have higher body fat, poorer glucose tolerance, and there are hypothalamic peptides that are different. So how much does this influence the potential for socioeconomic status transitions? There's a small body of literature on adoption as neonates or children, and individuals who lost income or gained income. And it turns out that in all of these three cases, there's a carryover from the prior SES status. So this is in a way makes me think that back to electron transitions, but there's no photons released by these transitions. Rich knows what I'm talking about. So there's an immense future that needs to be worked out of how we can intervene in people with different biological damage from their SES status. And essentially, I think the future of healthy brain aging is to minimize things that cause aging that I call gerogens for optimizing gene environment and development through aging. That is what I wanted to talk about. I'm open for questions.
There was a question in the chat from Nan. Do you want to repeat it?
Yes. I'm sorry. So if an older adult, low education older adult or middle-aged adult start learning new things, maybe in later their life, their dementia risk down a little.
I can't hear the question.
Tuck, maybe I can read it for you. If a low education older adult or middle-aged adult starts reading and learning new things, will they lower the dementia risk?
I think the answer is it makes no difference, because there's a different biological process that's underway in the brain, and what you read is of interest to you, but it's not going to change the biology of aging in the brain. Crossword puzzles don't help.
Are there other questions?
I have a question. if you learn and retain differently. For example, it's been shown, and I know you have a warm heart for music. I do too. And it turns out that even Alzheimer's and dementia don't affect the recollection of how to play the instrument or the lyrics, while you're forgetting, and it does affect other things. So it would seem that there seems to be some sort of difference in how you're retaining it, and it is a different part of the memory. So are there different parts of your memory that aren't affected or that are affected by this?
That's a terrific question, and it makes the point that our term Alzheimer's disease is probably no longer adequate, because some recent studies that did on large numbers, more than 1,000 people from middle age into later age, there were 20-year studies with brain imaging, shows that there's very poor correlation between the Alzheimer plaques and tangles and cognitive status. And it turns out that besides the amyloid and the neurofibrillary tangles, there are at least 4 other pathological aggregates of brain proteins that are present to varying degrees in individuals. And so it's, for some of us, and I'm on this side, it's like it's said for autism. If you've seen one case of autism, you've seen one case of autism. And I think the same thing could be said for, if you've seen one case of dementia, you've seen one case of dementia. So referring to your awareness that there's multiple domains of memory, there are people who can't speak, remember their names of their closest people, who can sit down and play something at the piano.
Or you can play the instrument while you're talking to somebody. They come up and start to talk to you, and you just keep playing because your brain knows how to do that. It's not that you,
it's just, the brain is an immense labyrinth to neurobiologists, even though we can name the parts.
I have 2 questions.
Yes, Karen.
One is general and the other is personal. The first general question is recently I attended a women's renewment event in which we were all sharing stories of aging. And one of the presenters was talking about her working with her mother, who had Parkinson's, went into dementia, and there's sort of a movement to say that dementia, Alzheimer's, is not a disease, but it's a disability. And I wondered if you had thoughts about that.
Well, this depends upon where the question comes from. It's a disease because it causes damage to the anatomical damage to the brain. Heart disease is a disease, and there's varying levels of disability depending upon the person and the state of the disease. So disability is a consequence of a disease, rather than the other way around. That's how I just at this moment think about it. But
okay, thank you very much. And then the personal question, my mother has very advanced dementia. I don't know if it's Alzheimer's. You don't know until the brain is dissected. I'm not doing that. And so, of course, I have a fear of that it's hereditary genetic. And you say, I know that crossword puzzles don't help because it's going back into memory, but taking a very difficult foreign language, would that kind of jumpstart parts of the brain at this age?
Once the synapses have are lost, you don't get them back.
But would new ones open?
Probably. I mean, the reality is that the level of mental activity doesn't seem to change the course of the disease once it's been gotten to a clinical stage.
Is there a way you can put the brakes on it? In other words, you can't recover what's gone.
Well, there are 3 drugs that are now in use, antibodies to the amyloid peptide, that all of them slow cognitive loss by about 25% or 30%. So there's 3 different, I'm not going to name them because I don't promote drugs, but they all have despite the benefit, about a third of the people who take them have an additional major problem, that they have brain swelling, edema, and hemorrhage, which then requires expensive and dangerous treatment. So that's where the state is. 5 years ago, we did not have those drugs. And of course, they're immensely interesting to all of us.
Is Xcopri one of them?
I don't recognize the name.
That's the trade name. I'd have to look at the drug name. Okay, it's an anti-seizure med, so I didn't know.
No, it's not one of them.
Okay, yeah.
The drugs that are in the market that are in this year are antibodies to the amyloid peptide that require 2 injections in a hospital a month, twice a month.
Okay, thank you.
Yeah.
Is one of them, one of the drugs recently approved by the FDA?
Yeah, 2 of them are recently approved.
Okay. I know they're controversial.
Well, they work, but they have side effects, and nobody knows how long they will work and whether they will work as a protective for people who are perfectly normal. I imagine there's going to be a big demand for people who are 50 who had a parent with Alzheimer's who want to be protective, and just think of how that's going to go.
Tuck, Richard Bergman, I want to congratulate you on a wonderful career and a wonderful talk, and we really appreciated it. And I think that any of us who's been in this game for a long time, it's a wonderful opportunity to look at the whole plethora of things that we've done. And you have, of course, a very great brain for going from one thing to another. So from the Bergmans over here, we really loved your talk, and I want to thank you.
Thank you. And I love every conversation we've had since the first. Okay, I've got to run to another meeting. I'll be receptive for any emails to me, and thank you for joining the conversation.
Well, let me echo Professor Bergman's comment. Congratulations on a stunning career, and may there be many more publications. And thank you very much for your talk today.
And thanks to USC for being so supportive for six different provosts and presidents. May it continue.
And if you're ready to jam, I've been playing for over 60 years, so I'll send you an email with contact.
Okay.
Okay, well, thank you all for coming to this. I just want to mention that the next Intellectual Journeys talk will be given by Sandy Sawchuk in the school of Viterbi School, and that will be in November. I can't remember the date offhand. I think it's November 11th, but there will be an ad coming out very soon about that, and please watch for it. So thank you all, and have a very good rest of the day.